My name is Nafisa Hamza, and I'm a junior at Tennessee State University. The project that I've been investigating is how signaling pathways are involved when Tributyltin-Induced Increases of Interleukin 6 are seen. So Tributyltin, which I'll be referring to as TBT, has many industrial uses. Because of its lipophilic character, it readily absorbs into organisms. So it's found in our environment in concentrations as high as 261 nanomolar. It is still toxic, and we use it in our everyday lives, so it's really important to investigate the effects it has in our immune system. Specifically, what I'm looking at is how TBT affects interleukin 6 in our immune system. Interleukin 6 is a cytokine, which is cell signaling protein. It helps with cell immune responsiveness as well as the regulation, mature responsiveness of other cell populations. So, it's really important to see how TBT affects interleukin 6. Well, first, we looked at how TBT affects the lymphocytes, and after 24-hour exposure of TBT, we've noticed that the secretion levels of interleukin 6 had little effect on it. However, the production of the IL-6 in our cells, which is the intercellular levels as well as the secretion, we noticed that there was an increase of interleukin 6 production at 24-hour exposure of TBT at all concentrations levels. So the next step was to figure out why there was an increase of production of interleukin 6 in our lymphocytes. So we looked at the MAP kinases, which are the reason which help regulate interleukin 6 in our body. Specifically, we looked at 3 MAPK signaling pathways: the ERK1/2 pathway, the P38 pathway, the J&K pathway. Okay, the results by ELISA and Western blot indicate that the P38 pathway, when the inhibitor was present, showed that there was an increase of production of interleukin 6, showing that TBT-induced increases might be reliant on the activation of this pathway, as well as the ERK1/2 pathway. The ERK1/2 pathway as well shows similar results. When the inhibitor was blocking the J&K pathway at the higher concentrations, like at 200 nanomolar of TBT, showed that the inhibitor did not block this pathway. However, at the lower concentrations of certain donors, it showed that there was a decrease, which shows that maybe it is using this pathway. For the NF-kappa B transcriptional activator, it showed that similar to the J&K pathway, at certain concentrations, it showed that the inhibitor did not affect it. We can conclude that TBT might be utilizing the ERK1/2 pathway as well as the P38 pathway. Thank you for coming by, and if you want to get in contact with me, you can check out my email. It's probably right here.